We're on to Lab 5, our first organ system, the integumentary system. And we'll begin here with the three general layers that we see here, and then we'll go back to the details. So we'll start over here. We have a model that's been labeled. So you have the image of the model with the numbering, and here on the next page, you'll have the answer key for it. So we'll start with the unlabeled model. And again, the three general layers, and then we'll go back to the details. So we can start at the surface. We have the epidermis. And we see the difference between thin and thick skin, but here we have the epidermis. And then now the second layer of the skin. Here's now where we have the dermis. And now moving beyond those two layers, here where you see all the yellow representing adipose, this is the area of the hypodermis or subcutaneous layer. So now going back to the details, we can start here in the area of the epidermis, and you can start to see the different colors. And the different colors represent the various strata of the epidermis. And we have it listed out here again, so the distinct strata. And the way that we'll take a look at that, we'll use this program here. So this Practice Anatomy Lab, a good resource, this program or website as well. And we'll start with the epidermis. And to see all five layers, we'll use the thick skin region. So this part here of the model in particular. And again, working with the color coding. So it's right at the surface, stratum corneum. And then you can see the major color difference on the model, representing that next layer, stratum lucidum. The next shade, stratum granulosum. And after that, we can see the color change, representing now the stratum spinosum. And then finally, the deep edge, the stratum basale. And we'll just go in reverse to see all of those layers. Now, related to the epidermis, we have what's called the epidermal ridges. The epidermal ridges are composed of the stratum basale and the stratum spinosum. And that's the area where the epidermis is literally protruding deeper into the skin. So all of these kind of downward projecting areas, and we'll go to the image to get a better view. But areas where the epidermis is protruding deeper into the skin, like right here, is a good example of a epidermal ridge. And now that leads us into the second layer of the dermis. And we'll start with the fact that the dermis has areas that protrude outwards towards the surface. So we can label it right in here. These portions of the dermis that protrude outward towards the surface, these are known as the dermal papillae. Or if you notice for this area of the model, the epidermis is removed, and that's all you see are just dermal papillae, just protruding outwards towards the surface. So we'll continue along now with the dermis, which is divided into a papillary layer that contains the dermal papillae and the reticular layer. So going back to the model here. The dermis has two subdivisions, and the dividing lines are right about here by the blood vessels. We'll just draw this in. So just this very thin superficial area of the dermis is called the papillary layer. And this is the area that, of course, contains the protruding dermal papillae. Whereas the deeper majority of the dermis, this is called the reticular layer. It's now within the papillary layer. Not only do we have the dermal papillae, but this nerve ending appearing here as the white structure. These are known as tactile corpuscles, or another name is Meissner's corpuscle. So a type of touch receptor. So now we'll continue with the dermis, moving beyond to the reticular layer. And then again, we've already pointed out the hypodermis. And the hypodermis, of course, containing large clusters of adipose tissue. This final trait, now another nerve ending, a lamellated corpuscle. On this model, it's seen in the hypodermis, and that's the feature seen right here. So another type of sensory touch receptor, a lamellated corpuscle or Pacinian corpuscle. So now continuing with the glandular structures of the integument, sebaceous glands and two types of sweat glands. So to begin, this model has three different types of skin, three different segments of skin on it. There's a portion here that's representing the skin on the scalp. A portion here is representing skin from the axilla, the underarm. And this last portion here is representing an area of thick skin, such as the palm of the hand, the sole of the foot. So we'll start with the sweat glands, the ones here shown in white. So you notice these are spread throughout all three of these different skin samples. So this is a very common eccrine sweat gland, or also known as merocrine sweat gland. So eccrine, or also known as merocrine sweat gland. Here in the axillary region, that is one location of the very large, now apocrine sweat gland. And then lastly, a sebaceous gland. Sebaceous glands are always connected to a hair follicle. And so we see these here now in this blue color. And here's one that's now sectioned open, a sebaceous gland. And we see how both of those are connected right into a hair follicle, which is always their position. And moving on to the hair structure, there's the follicle, the organ that develops hair. We have the erector pili muscle that's connected to the follicle, as well as that sebaceous gland we just pointed out, and the root shaft and the hair papillae. All right, so to begin, the hair follicle here on these models are represented by the different shades, the purple, the pink shades all together as the hair follicle. And let's get a better color we can see. So here's a hair follicle that's been sectioned open, or a complete hair follicle. At the very base of the hair follicle, we can see right here, you notice the blood vessels connecting right in. That's the area of the hair papillae. The hair root, in this case it's brown hair. The hair root's the deep internal portion of the hair. And the hair shaft is the exposed superficial portion. We've already pointed out the sebaceous gland. And we see now this muscle that's running at an angle coming off of the hair follicle. Here's now the location of that erector pili muscle. Surface, the epidermis. We would zoom in closer to see the individual strata. So now deep to that, we have the dermis. And it even goes far enough to divide, there we go, papillary layer, superficial, and then the deeper reticular layer of the dermis. That's the two major layers for the skin. And now deep to that, the underlying cushion of hypodermis. Let's do the nerve endings. There's the more superficial Meissner's corpuscle, also known as tactile corpuscle, and the deeper Pacinian or lamellated corpuscle. We'll do the sweat glands.
Pacinian corpuscle.
So there's what we see as the merocrine or eccrine sweat glands. Not seeing the labeling for that. And the much larger apocrine sweat gland. Sudoriferous is another term for sweat, sudoriferous gland or sweat gland, so apocrine sweat gland. The hair structure, we have the hair follicle. There's now the very deep base there of the hair papilla. The internal hair root, and exposed at the surface, the hair shaft. Connected to the hair's sebaceous gland, as well as the erector pili muscle. All right, so we have several features seen here on the skin. We have a chart here now comparing those three different sections of the skin model, the three different types of skin. So I'll let you work on this yourself to determine, is hair located on the scalp, the armpit, thick skin? So you could check that off as you're working through this section. So you'll see that comparison between different types of skin, what features are present. And the final part for the gross anatomy here is this figure of the fingernail. And it's this textbook figure seen here. And what we want to point out are the different items. So we'll start with this view. We have what's called the eponychium. So this skin here, right on the proximal end of the nail. And then once we reach the nail here, we have this region of the nail called lanula. It's that more opaque region there, as opposed to seeing the pink coloration from capillaries. And then moving over to here is another view of that eponychium and the location of the lanula. The skin that's now growing under the distal end of the fingernail, so the hyponychium. And the very base of the nail itself, that's known as the hair root. So that's all from this figure right here in the textbook. And the final portion for this lab is now the histology. And histology, of course, is posted on our Canvas page, and we'll work through the slides here as well. So again, like the model, we'll start with the three general layers, and then we'll fill in the details. So starting with this first micrograph here, we'll take a look at the three general layers. So we have this darkened, stained edge. That's where we're seeing the epidermis. We have this pink staining here, the connective tissue layer, the collagen, and the dermis. And then you can already start to see the adipose tissue. You should be able to recognize that sample, and that's where we have the hypodermis. So now for the details within, for the epidermis, you want to see the stratified squamous epithelium once again. So we have an alternate skin sample we'll go to and zoomed in. So it's from this area upwards is the stratified squamous epithelium. It's the dry type. And we're seeing that right here at the surface of the skin, right at the border of the sample. Or as another view zoomed out again. So we're able to see the epidermis composed of the stratified squamous epithelium, the keratinized or dry type. And then as we move deeper in now, here's where we're seeing the area of the dermis. So now to point out details within the dermal layer, we want to be able to see the hair follicle, the dermal papillae, erector pili muscle, sebaceous glands. All right, so here we are in the area of the dermis, and we'll scroll through a couple of different slides to see these features. So we have the dermal layer, of course, and here we see a couple examples of some hair follicles, just a small segment. You'd have to imagine this follicle is running at an angle coming, say, out the screen. So we just see that small segment of the hair follicle with some of the hair roots still within. Here we have now a better sample. Once again, we're able to see a hair follicle, so this darker stained oval region. And now we see connected to it and running at an angle. Here's the appearance of that erector pili muscle. Pretty good sample there, really good sample. I'll put APM, erector pili muscle. And also you notice right here, we see that cluster of cells. This is now an example of that connected sebaceous gland. So it's both the erector pili muscle and the sebaceous gland. We see both closely associated and attached to this individual hair follicle. Or just another view of a hair follicle. This time we don't see that remaining hair within, but we can see that sample of smooth muscle tissue, making that an erector pili muscle. And a little segment of a sebaceous gland seen here. Now this is a really good sample here. We see much more length of the hair follicle. So this cuts right in the plane to follow the length of the hair follicle. And we once again can see some of that hair that's still running within that hair root. And another good example of a sebaceous gland. And you can really see it leads right into that hair follicle. And this is now just a much closer magnification of segments of a sebaceous gland. And once again, that associated hair follicle. So another feature of the dermis are now the dermal papillae. So here we are in the area of the dermal layer. And we can see those protruding segments. This is a sample of thick skin. If you notice the prominent epidermis. And we're using the thick skin sample because the dermal papillae are much more prominent on thick skin. So now we're ready to transition from the dermal layer. And again, you can start to see the adipose tissue. And we know we're in the hypodermis. So that underlying cushion of adipose tissue. And even deep to that, we're starting to see now some muscle. So that's our histology, and that finishes up our Lab 5 and this first unit of the course. As a reminder, this next page here is just a helpful review of the various body cavities that we discussed in Lab 1, going through each cavity and the membranes that are present, as well as the organs. So for example, pericardial cavity has the heart, and the membranes located in this area are the visceral and parietal pericardium, a list of organs in the mediastinum, the pleural cavity, thoracic cavity, and so on. But again, you do not have to memorize every organ on this list. This is just as a reference for Lab 1, the general placement of organs in the cavities, and also a review from the first day, overall general functions of the 11 organ systems. But that was covered, of course, in the first lecture.